Biology has rules. They are taught early, they are printed in textbooks, and they are correct almost all of the time. Life needs liquid water. Cells are microscopic. Every complex organism breathes oxygen. Everything dies. An individual is one organism. All food chains begin with sunlight. Every one of those statements is wrong, and in each case we know precisely what is wrong with it, because we have the organism in a jar. There is an animal that survives being heated past the boiling point of water, frozen to a degree above absolute zero, exposed to the vacuum of space, and dosed with 1,000 times the radiation that would kill you. There is a bacterium you can see without a microscope, longer than a housefly, that keeps its DNA in membrane-bound compartments it should not have. There is an animal with no mitochondrial genome at all, which means it does not breathe. There is a jellyfish that ages backward, reversing its own development and starting over, apparently without limit. There is an ecosystem 2 miles underground powered by the radioactive decay of rock, in which a single species is the entire food web. None of these are hypothetical. All of them have been collected, sequenced, and described. The interesting thing is not that the exceptions exist. It is what they have in common, which is where we found them. Almost every organism in this video was discovered in the last 50 years, most in the last 20, and nearly all of them in places we had previously classified as uninhabitable and therefore not searched. The rules were not wrong because biology is unpredictable. The rules were wrong because we wrote them from a sample consisting almost entirely of the warm, wet, well-lit surface of one planet, and then assumed the sample was the whole. Tonight, we are going to work through those rules one at a time and meet the organism that breaks each one. And by the end, the pattern in the exceptions is going to turn out to be more interesting than any individual exception, because it says something specific about how much of this planet we have not looked at. Let's start with the first rule, and with an animal that survives conditions that do not exist anywhere on Earth. Welcome to Omni, where science meets story and curiosity leads the way. Every week we dive into the hidden patterns of life, the universe, and everything in between. If curiosity drives you too, leave a thought in the comments, hit like, and subscribe to keep exploring with us. The rule is that life requires liquid water and a moderate range of temperature and pressure. The organism that breaks it is about 0.5 millimeter long, has 8 legs with claws, walks with a gait that has been compared to a bear's, and is one of the most durable animals ever described. Tardigrades were first observed in 1773 by a German pastor named Johann Goes, who called them Kleiner Wasserbär, little water bear. The Italian biologist Lazzaro Spallanzani gave them the name we use now, meaning slow stepper. There are around 1,500 described species, and they are everywhere. Moss, lichen, leaf litter, sediment, glaciers, the deep sea, and probably the gutter outside your window. Under normal conditions, a tardigrade is unremarkable. It needs a film of water around it to be active. It feeds by piercing plant cells or eating small invertebrates, and it lives a few months. The remarkable behavior begins when its environment dries out. Rather than dying, the tardigrade retracts its legs, pulls its head in, contracts into a barrel shape, and expels almost all of the water in its body. In this state, called a ton, its water content can drop below 3% of normal. Its metabolism becomes undetectable, sometimes measured at less than 1/10,000th of the normal rate. In this state, the tardigrade is not doing anything that would ordinarily qualify as being alive. It is not metabolizing, not moving, not reproducing, not responding. And it can stay that way for years, and in some documented cases for decades. Add water, and within minutes to hours, depending on how long it has been dry, the animal reinflates and walks away. What a ton can survive has been tested extensively, and the numbers are worth stating precisely because they are usually exaggerated. Temperature. Tardigrade tons have survived exposure to temperatures above 150 degrees Celsius for brief periods, and cooling to within a fraction of a degree of absolute zero. In 2020, a paper by Ricardo Neves and colleagues did an important corrective, demonstrating that tolerance to high temperature is much lower in active tardigrades, and that even desiccated specimens have limits that depend heavily on exposure duration. The extremes are real, but they are brief exposure limits, not comfortable conditions. Pressure. Tons have survived pressures around 6,000 atmospheres, which is roughly 6 times the pressure at the bottom of the Mariana Trench. Radiation. Tardigrades survive doses in the range of several thousand grays. A dose of around 5 grays is lethal to a human. The dose is not selective for tardigrades, which is the important detail. It damages their DNA too. They survive because they repair it, vacuum. In 2007, the European Space Agency flew an experiment called TARDIS on the Photon M3 mission, exposing tardigrades to open space in low Earth orbit for 10 days. They were subjected to vacuum, and in some groups, unfiltered solar ultraviolet radiation. A team led by Ingemar Jonsson published the results in Current Biology in 2008. Animals exposed to vacuum alone survived at high rates and went on to reproduce normally. Those additionally exposed to the full solar ultraviolet spectrum fared much worse, but some survived even that. The mechanisms are now partly understood, and they are more interesting than the survival itself. The first is trehalose, a sugar that some tardigrade species accumulate during drying. As water leaves the cell, trehalose forms a glass-like amorphous solid that physically holds proteins and membranes in position, preventing them from unfolding or fusing. This is called vitrification, and it is used by a range of desiccation-tolerant organisms, including some yeasts, plant seeds, and brine shrimp. But some tardigrade species make very little trehalose, which meant something else had to be going on. That something turned out to be a class of proteins found nowhere else, identified in 2017 by Thomas Boothby and colleagues, called tardigrade-specific intrinsically disordered proteins. These proteins have no fixed structure in solution, which is unusual because most proteins fold into a specific shape to function. As the cell dries, these proteins vitrify, forming a glassy matrix that appears to serve the same protective role as trehalose. Expressing them in yeast and bacteria confers desiccation tolerance on organisms that did not have it. The radiation tolerance has a separate explanation, and it came out of the genome. In 2016, a Japanese team led by Takuma Hashimoto and Takekazu Kunieda sequenced the genome of Ramazotius varioornatus, one of the more tolerant species, and identified a protein they named DSUP, short for damage suppressor. DSUP binds to DNA and appears to physically shield it from hydroxyl radicals, which are the primary agents of radiation damage since most radiation injury is mediated by water breaking apart rather than by direct hits. The demonstration was direct. They inserted the DSUP gene into cultured human cells. Those cells showed roughly 40% less DNA damage after X-ray exposure than unmodified controls. A gene from a half-millimeter animal transferred into human tissue culture made human DNA measurably more radiation resistant. Tardigrades are not alone in this territory, and some of their neighbors are more extreme in specific dimensions. Deinococcus radiodurans is a bacterium that was discovered in 1956 in a can of ground meat that had been sterilized with gamma radiation and spoiled anyway. It survives doses around 5,000 grays with no loss of viability, and it can survive 15,000 grays with reduced survival. That dose shatters its chromosome into hundreds of fragments. It reassembles them. Over roughly 3 hours, the organism reconstructs its genome from the pieces using a process involving extended synthesis-dependent strand annealing and the fact that it carries multiple copies of its genome that can serve as templates for each other. The reconstruction is accurate. The puzzle is why it evolved this, since natural radiation at that level does not exist anywhere on Earth's surface and never has. The leading explanation is that the radiation resistance is a side effect of adaptation to desiccation, which produces very similar DNA damage. The organism evolved to survive drying out, and radiation resistance came along for free. For heat, the record holder is an archaeon called Geogemma barosi, informally strain 121, isolated from a hydrothermal vent in the northeast Pacific. It grows at 121 degrees Celsius, which is the temperature used in autoclaves to sterilize medical equipment, and it survives brief exposure to 130. It is kept liquid by pressure. For depth, there is Halocephalobus mephisto, a nematode named after the demon in Faust, described in 2011 from water in South African gold mines at depths of over 3 kilometers. Before that discovery, the deep subsurface was assumed to be a microbial domain exclusively. Finding a multicellular animal down there, in water that had been isolated for thousands of years, changed the assumed limits for animal life. The rule is that cells are microscopic and that bacterial cells are the smallest of all. The reason is diffusion. A cell has to move molecules around inside itself, and in the absence of an active transport system, molecules move by random thermal motion. The time it takes something to diffuse a given distance scales with the square of that distance. Double the size of the cell, and the transit time quadruples. Past a certain point, the interior cannot be supplied fast enough, and the cell starves in the middle. This is why bacteria are typically 1 to 5 microns long, and why eukaryotic cells, which do have active transport along a cytoskeleton, top out around 100 microns for most types. In 2022, a paper in Science by Jean-Marie Volland and colleagues described a bacterium that is up to 2 centimeters long. Theomargarita magnifica was collected from sunken mangrove leaves in the shallow waters of Guadeloupe in the French Caribbean. It had actually been noticed years earlier by Olivier Gros, who assumed he was looking at some kind of fungus or small eukaryote, because it is a visible white filament that you can pick up with tweezers. It is a single cell, not a filament of many cells stacked end to end, which is common in bacteria, but one continuous cell with one continuous membrane. The average length is around 1 centimeter, with the largest specimens near 2. That is 50 times longer than any previously known bacterium, and roughly 5,000 times the length of a typical one. In terms of relative scale, the comparison offered by the authors was that finding it is like encountering a human as tall as Mount Everest. It solves the diffusion problem in 2 ways. First, most of the interior volume is taken up by a large central vacuole filled with fluid, which pushes the metabolically active cytoplasm into a thin layer just under the membrane. So the cell is enormous, but the living part of it is a thin shell, and nothing has to diffuse very far. Second, and this is the finding that made the paper significant beyond the size record, the DNA is not free in the cytoplasm. One of the defining textbook distinctions between bacteria and eukaryotes is that bacteria have no membrane-bound organelles and no nucleus. Their DNA floats in the cytoplasm. Theomargarita magnifica has hundreds of thousands of copies of its genome distributed throughout the cell, and each copy is contained within a membrane-bound compartment along with ribosomes. The authors named these compartments pepins. Each one appears to be a self-contained unit of transcription and translation. That is not a nucleus in the eukaryotic sense, and the authors were careful about that. But it is compartmentalized DNA with associated protein synthesis machinery inside a bacterium, which is a category of thing that wasn't supposed to exist. There are large single cells among eukaryotes too, and one of them is a plant you can buy at an aquarium store. Caulerpa is a genus of green algae. Some species grow to 1 meter or more in length, with structures that look exactly like roots, a stem, and fern-like fronds. There are no cell walls dividing it internally. The entire organism is one continuous cytoplasm containing many thousands of nuclei, a condition called koinocytic. If you cut a caulerpa frond, cytoplasm flows out, and the organism seals the wound by forming a plug. How a single cell produces differentiated structures that look like organs, with no compartmentalization to keep the instructions separate, remains an open question. Something is establishing positional information across a meter of shared cytoplasm, and the largest single cell most people have handled is in the kitchen. The yolk of a bird's egg is a single cell, and an ostrich yolk is around 8 centimeters across. There is a related case that is worth adding because it behaves in a way that a single cell has no business behaving. Physarum polycephalum is a slime mold, which is neither a plant, an animal, nor a fungus. In its feeding stage, it is a plasmodium, a single cell containing millions of nuclei, spreading across a surface as a yellow network of veins that can cover a square meter. It has no nervous system and no brain. It has been shown, repeatedly, to solve problems that look like they require one. In 2000, Toshiyuki Nakagaki published a paper in Nature showing that a plasmodium placed in a maze with food at 2 exits would, over several hours, withdraw from the dead ends and leave a single connecting tube along the shortest path between the 2 food sources. In 2010, Nakagaki's group published a follow-up in Science that is more striking. They placed oat flakes on a surface in the positions of the major cities around Tokyo, scaled to a map, and let a plasmodium grow from the position of Tokyo itself. The network it produced over about 26 hours had comparable total length, fault tolerance, and transport efficiency to the actual Tokyo rail network, which was designed by engineers over decades. The mechanism is not planning. It is a feedback rule operating locally. Tubes carrying more flow are reinforced and thicken. Tubes carrying less are reabsorbed, and the cell's internal fluid oscillates back and forth on a rhythm of roughly 2 minutes, distributing material. The global solution emerges from a local rule applied everywhere at once. Physarum has also been shown to habituate to a repeated harmless stimulus and to recover that response after a rest period, which meets some of the technical criteria for a simple form of learning, and work by Audrey de Sautour's group has demonstrated that this learned state can be transferred to another plasmodium by fusing the 2 together. A single cell with many nuclei and no neurons that solves network optimization problems and can pass a learned behavior to another cell by merging with it. The rule is that all animals require oxygen and that all eukaryotic cells contain mitochondria. Mitochondria are the organelles that perform oxidative phosphorylation, using oxygen to extract energy from food. They descend from a bacterium engulfed by an ancestral cell roughly 1.5 to 2 billion years ago, and they retain a small remnant genome of their own. For a long time, the mitochondrion was considered non-negotiable. Every eukaryote had one, even the ones that live without oxygen, which retain reduced versions called hydrogenosomes, or mitosomes that no longer produce energy but still perform other essential functions, particularly the assembly of iron-sulfur clusters that many proteins require. In 2016, a team led by Anna Karnkowska published a paper in Current Biology describing monococomanoids, a single-celled organism found in the gut of a chinchilla which has no mitochondrion of any kind, not a reduced one, none. The genome contains no genes for any mitochondrial function. It had solved the iron-sulfur cluster problem by acquiring an entirely different system, apparently by horizontal gene transfer from a bacterium that performs the same job in the cytoplasm. Having that alternative in place, it was able to discard the organelle completely. That was the first eukaryote known to have done so. But it is a single-celled organism, and the rule about animals held for 4 more years. In 2020, a team led by Diana Yahalomi and Dorothée Huchon at Tel Aviv University published a paper in the Proceedings of the National Academy of Sciences on a parasite called Heneguia salminicola, which lives in the muscle tissue of salmon and produces the white cysts sometimes called tapioca disease. Heneguia is a mixozoan, and mixozoans are one of the strangest lineages in the animal kingdom. They are cnidarians, meaning they belong to the same phylum as jellyfish, corals, and sea anemones. Over evolutionary time, as parasites, they have been reduced to a handful of cells. They still possess structures called polar capsules that are unmistakably derived from the stinging cells of their free-living relatives, repurposed for attaching to hosts. The team sequenced the organism and found no mitochondrial genome. They confirmed it by staining for mitochondrial DNA, which produced no signal, and by looking for nuclear genes involved in mitochondrial replication and transcription, which were absent. This is an animal, in the formal taxonomic sense, that has abandoned aerobic respiration entirely. How it obtains energy is not fully established. The likely answer is that it takes it directly from its host, which would make it the animal equivalent of a parasite that has given up on making its own energy. There is also a contested case involving larger animals. In 2010, a team led by Roberto Danovaro reported finding loriciferans, tiny multicellular animals under a millimeter long, living in the permanently anoxic hypersaline basins of the Mediterranean deep sea, and reported that they appeared to be metabolically active and possessed hydrogenosome-like organelles rather than mitochondria. Subsequent work has questioned whether those animals were genuinely living there or had drifted in as carcasses, and the case remains unsettled. I include it because it is frequently cited as established, and it is not. The rule is that everything dies, and more specifically that every organism ages, accumulating damage until it fails. The counterexample most people have heard of is a jellyfish about 4 millimeters across. Turritopsis dohonii has the standard cnidarian life cycle in one direction. A fertilized egg becomes a swimming larva, the larva settles and becomes a polyp attached to the seafloor. The polyp buds off small jellyfish, and the jellyfish grow, reproduce sexually, and die. What Turritopsis can do is run that sequence backward. An adult medusa, when it is starving, injured, or otherwise stressed, can contract its bell, absorb its own tentacles, sink to the bottom, and reorganize its body into a cyst, which then develops into a polyp. The polyp buds off new medusae, which are genetically identical to the original. The critical part is what happens at the cellular level. This is not regeneration from stem cells, it is transdifferentiation, in which already specialized cells change directly into other specialized types. Muscle cells become nerve cells. Cells that had committed to one fate revert and commit to another. In most animals, differentiation is close to a one-way process, and the discovery that mature cells can be reprogrammed at all in the laboratory won Shinya Yamanaka a Nobel Prize in 2012. Turritopsis does it by itself, on demand, as a stress response. The phenomenon was first described in the 1990s by researchers including Ferdinando Boero and Stefano Piraino working in the Mediterranean. In 2022, a team led by Maria Pascual-Torna and Carlos Lopez-Otin at the University of Oviedo published a genome comparison in the Proceedings of the National Academy of Sciences between Turritopsis dohonii and Turritopsis rubra, a close relative that cannot perform the reversal. They found differences in genes governing DNA repair, telomere maintenance, and the regulation of cellular differentiation, including duplications of certain repair genes in the immortal species. The important qualification, which is almost always omitted, is that individual Turritopsis are eaten and killed constantly, and there is no evidence any individual has actually persisted indefinitely. What has been demonstrated is that the animal has no known internal limit on the number of cycles, not that any particular one has lived forever. For an animal that simply refuses to age in the conventional sense, the better example is Hydra. Hydra is a small freshwater polyp, a few millimeters long, and its body is composed almost entirely of stem cells that divide continuously. The animal replaces its entire cell population roughly every 20 days. It is, in effect, continuously rebuilt. Daniel Martinez published a study in 1998 that tracked Hydra for 4 years and found no increase in mortality rate with age and no decline in reproductive output. That is the technical definition of negligible senescence. A study by Ralph Schabel and colleagues in 2015 extended the observation to 8 years with the same result, and calculated that under the observed mortality rate, 5% of a hydra population could still be alive after well over 1,000 years. Then there are organisms that simply persist. Pando is a stand of quaking aspen in Utah. It appears to be a forest of roughly 40,000 individual trees spread over about 43 hectares. Genetic testing established that they are all one organism, a single male clone connected by a shared root system, with each trunk being a stem rather than an individual tree. Age estimates for Pando vary widely and are genuinely uncertain, ranging from several thousand years to as much as 80,000, because there is no method for dating a clonal root system directly. What is well established is that Pando is currently in trouble. Research led by Paul Rogers has documented that browsing by deer and elk is preventing new stems from surviving to maturity, so the clone is aging without replacement. In the Mediterranean, there is a seagrass, Posidonia oceanica, and a clone in the waters near Formentera has been estimated by genetic analysis to be somewhere between 80,000 and 200,000 years old, spanning 15 kilometers. For individual non-clonal animals, the records are held by the cold and the slow. The ocean quahog is a clam. A specimen collected off Iceland in 2006 was aged by counting annual growth bands in its shell, a method that has been validated against radiocarbon dating. The count came to 507 years. It was born around 1499. It was named Ming, for the dynasty ruling China at the time, and it was killed by the process of being opened for study. The Greenland shark holds the vertebrate record. In 2016, a team led by Julius Nielsen published a study in Science using radiocarbon dating of the eland's nucleus, which is metabolically inert and formed before birth, effectively preserving a chemical timestamp. The largest sharks in the study came out at around 392 years, with an uncertainty range of plus or minus 120. Even at the low end, these animals live longer than any other vertebrate known, and they do not reach sexual maturity until around 150 years old. Among trees, the oldest verified non-clonal individual is a Great Basin bristlecone pine in the White Mountains of California, aged by ring count at over 4,800 years. Its location is kept secret because a previous record holder was cut down in 1964 by a graduate student whose coring tool broke with the permission of the Forest Service, and only afterward found to be the oldest known tree on Earth. Bristlecones survive by growing extremely slowly in hostile conditions, producing dense resinous wood that resists rot and insects, and by allowing most of the trunk to die while a narrow strip of living tissue continues to supply a few branches. Then there are the sponges. Glass sponges in the Antarctic and the deep Pacific grow so slowly that age estimates for large specimens run into the thousands of years, with one frequently cited estimate for a Monohaphis chuni specimen placing it at around 11,000 years, though the dating methods for these animals carry substantial uncertainty and the figure is debated. At the far end of the longevity question, where it stops being about aging and becomes about time itself, there is the permafrost and the salt. In 2018, researchers reported reviving microorganisms from Siberian permafrost samples hundreds of thousands of years old. Older claims exist, and they are contested. In 2000, a group led by Russell Vreeland reported culturing a bacterium from a fluid inclusion inside a salt crystal from a formation dated to around 250 million years, which would make it older than the dinosaurs. The result has never been convincingly replicated, and the most likely explanation offered by critics is contamination with modern organisms, which is extremely difficult to exclude in this kind of work. I mention it because it is a good example of how this field polices itself. The claim was extraordinary. It was published in Nature. It received enormous attention. And the community response was to attempt replication, fail, and move on without ever quite closing the file. The rule is that an individual organism is one organism. This one dissolves faster than any of the others once you press on it. Start with something you have walked past thousands of times. Lichen is the crusty growth on rocks, gravestones, tree bark, and roofs. It covers something on the order of 6% of Earth's land surface, and it grows in places where essentially nothing else does, including bare rock in Antarctica. For most of the 19th century, lichens were classified as plants. In 1867, a Swiss botanist named Simon Schwendener proposed that a lichen is not one organism, but two, a fungus and an alga living together, with the fungus providing structure and mineral capture, and the alga providing sugar through photosynthesis. The proposal was rejected loudly by most of the botanical establishment. The idea that a single visible organism could be a partnership between two kingdoms was considered absurd. Schwendener was eventually vindicated, and lichen became the standard textbook example of symbiosis. Then, in 2016, Toby Sprabilla and colleagues published a paper in Science that broke the textbook version. They had been trying to explain why two lichens with apparently identical fungal and algal partners could have completely different appearances and chemistry, one producing a toxic compound and one not. Standard sequencing of the known partners showed no difference that could account for it, so they sequenced everything present, without assuming what they would find, and they found a third organism, a Basidiomycete yeast, embedded in the outer layer of the lichen, present in both, but at different abundances. It had been overlooked for 150 years because nobody had looked for a partner they did not already expect. Subsequent work has found that many lichens contain additional fungi, bacterial communities that appear to be consistent partners rather than contaminants, and sometimes multiple algal species. The lichen is not a partnership between two organisms, it is a small ecosystem with a shape. For a version of this that is even harder to pass, consider Mixotricha paradoxa. It is a single-celled protist that lives in the gut of the Australian termite, where it helps digest wood. Under a microscope, it appears to be covered in cilia, the small hair-like structures many single-celled organisms use to swim. They are not cilia. They are around 250,000 bacteria of a type called spirochetes, attached to the surface of the cell in a regular arrangement, beating in coordination to propel it. The protist does not swim. It is carried by an organized crowd. There are further bacteria attached alongside the spirochetes, serving as anchoring structures. There are two more distinct bacterial species living inside the cell itself, plus the mitochondrial descendants that every eukaryote carries, except that in this case, they have been reduced to hydrogenosomes because the termite gut has no oxygen. The species name Paradoxa was well chosen. It is at least 5 organisms in a trench coat, and it lives inside a termite that cannot digest wood without it, in a colony that functions as a superorganism. Then there is the animal that is not an animal, but a fleet of them. The Portuguese man-o'-war looks like a jellyfish with a gas-filled float and long trailing tentacles. It is a siphonophore, and it is a colony of genetically identical but structurally distinct zooids, each derived from the same fertilized egg but developed into a different specialized form. Some zooids form the float. Others are the tentacles that capture prey. Others are digestive polyps. Others are reproductive. None of them can survive independently, and none of them is the animal. The organism exists only as the assembly. Siphonophores also include the longest animals ever recorded. In 2020, a Schmidt Ocean Institute expedition off Western Australia filmed an Apolemia siphonophore in a feeding spiral estimated at over 40 meters in outer ring circumference, with the animal itself likely exceeding the length of a blue whale. And there is the animal that stole photosynthesis. Elysia chlorotica is a sea slug on the eastern coast of North America. It is bright green and shaped like a leaf. As a juvenile, it feeds on a particular alga, and rather than digesting the chloroplasts, it retains them in the cells lining its digestive system. The chloroplasts keep working. The slug can then survive for months on sunlight alone without eating. This is called kleptoplasty, and there is a genuine ongoing scientific argument about how the slug maintains chloroplasts that require constant protein resupply from the algal nucleus, which the slug does not have. Early claims of horizontal gene transfer from alga to slug have not held up well under subsequent genome sequencing, and the mechanism remains unresolved. What is not in dispute is the observation. There is an animal that photosynthesizes, and it acquired the ability by eating something and keeping the parts. There is one more case in this category that inverts the usual direction of the relationship. Corals are animals, cnidarians, related to jellyfish and anemones. A reef-building coral is a colony of small polyps sitting in a limestone skeleton they secrete themselves, and each polyp has tentacles and stinging cells and can capture plankton. But the great majority of a coral's energy, typically cited at around 90%, does not come from hunting. It comes from single-celled algae called zooxanthellae living inside the coral's own cells in densities on the order of 1 million per square centimeter of tissue. The algae photosynthesize and pass sugars to the animal. The animal supplies carbon dioxide, nitrogen compounds, and a protected position in the light. That relationship is why coral reefs exist in tropical waters that are otherwise nutrient-poor, sometimes described as marine deserts. The reef is not living off the surrounding water. It is running an internal agricultural system and recycling nutrients so efficiently that it builds the largest biological structures on the planet in water that could not otherwise support them. It is also why bleaching happens. When water temperature rises even a degree or two above the local seasonal maximum for a sustained period, the relationship breaks down and the coral expels its algae, turning white and losing its primary energy source. If conditions return to normal quickly enough, the coral can reacquire algae and recover. If they do not, it starves. An animal that has outsourced its metabolism to a partner is an animal that can be killed by breaking the partnership, without anything having to attack it directly. The rule is that all food chains begin with sunlight. This one was overturned in a single afternoon in 1977, and it is probably the most consequential discovery in the history of biological oceanography. The submersible Alvin was working on the Galapagos Rift, about 2.5 kilometers down, on a geological expedition. The team was looking for hydrothermal vents to study the chemistry of seawater circulating through ocean crust. They were not expecting biology. The deep sea floor was understood to be a desert, sparsely populated by scavengers living on the slow rain of organic material sinking from the sunlit surface. What they found around the vents was a dense community. Clams 30 centimeters across, crabs, mussels, and tube worms, some over 2 meters long, in white tubes with bright red plumes, packed together in stands. There was no light. There was no possibility of light. Photosynthesis was not involved anywhere in the system. The energy source turned out to be chemical. Water percolating through hot rock near the spreading center becomes superheated and chemically reduced, and when it emerges, it is loaded with hydrogen sulfide. Bacteria at the vents oxidize that sulfide and use the released energy to fix carbon dioxide into organic molecules, which is chemosynthesis rather than photosynthesis. The tube worm, Riftia pachyptila, took this further than anyone expected. An adult Riftia has no mouth, no gut, and no anus. It cannot eat. Its body is largely filled with an organ called the trophosome, which is packed with symbiotic bacteria. The red plume is red because it is full of hemoglobin, and Riftia's hemoglobin carries both oxygen and hydrogen sulfide simultaneously, which is unusual because sulfide is normally a potent poison to oxygen-carrying molecules. It transports both down to the bacteria in the trophosome, which combine them to generate energy and produce organic compounds that the worm absorbs. The larvae do have a mouth. They use it once to acquire their bacteria from the surrounding water, and then the digestive system degenerates permanently. Riftia is also one of the fastest-growing invertebrates known, capable of extending well over a meter in under 2 years, which is remarkable for an animal in near-freezing water living on rock chemistry. Since 1977, chemosynthetic ecosystems have been found at cold seeps, at whale falls, in sediments, and throughout the deep subsurface. And the deeper implication took longer to sink in, which is that the biosphere is not a thin film on the surface. Estimates of the deep subsurface biosphere, meaning life in rock and sediment below the seafloor and below continental surfaces, place it at a substantial fraction of Earth's total microbial biomass. The Deep Carbon Observatory, a 10-year international program that reported in 2018, estimated the deep subsurface contains somewhere in the range of 15 to 23 billion tons of carbon in living organisms, and that the volume of habitable subsurface is roughly twice the volume of all the oceans. Much of that life operates at metabolic rates so slow they are difficult to comprehend. Some subsurface microbes have estimated generation times measured in centuries or millennia. They are not dormant. They are alive and metabolizing, just at a pace where a single cell division might take longer than recorded human history. And there is one system down there that is a single species ecosystem. Candidatus desulphurudis odax viator was described in 2008 from water 2.8 kilometers below the surface in the Mponeng gold mine in South Africa. The name comes from a Latin phrase in Jules Verne's Journey to the Center of the Earth, meaning bold traveler. The water it lives in has been isolated from the surface for millions of years. When the community was sequenced, it consisted of essentially one organism. Over 99% of the DNA recovered came from this single species. Everywhere else on Earth, ecosystems are communities. This one is not. The organism fixes its own carbon, fixes its own nitrogen, and gets its energy from sulfate reduction using hydrogen. And the hydrogen comes from radiolysis, meaning it is produced by the radioactive decay of uranium and thorium in the surrounding rock, splitting water molecules apart. It is a complete, self-sufficient, independent biosphere powered by radioactivity, consisting of one species that has been running in the dark for a very long time with no input from the surface world whatsoever. Which is, incidentally, the best model anyone has for what life might look like inside a body like Europa or Enceladus. There is one more site worth mentioning, because it may be the closest thing we have to a picture of where all of this started. In 2000, a research expedition on the mid-Atlantic ridge discovered a vent field unlike the black smokers found in 1977. It sits on a mountain of exposed mantle rock about 15 kilometers off the ridge axis, and it consists of carbonate towers up to 60 meters tall, pale and spired. The team named it the Lost City. Lost City is not volcanic. Its chemistry comes from a reaction called serpentinization, in which seawater reacts with olivine in mantle rock. The reaction produces heat without magma, and it generates hydrogen and methane in large quantities, along with fluids that are strongly alkaline, around pH 9 to 11, and warm rather than scalding, typically 40 to 90 degrees Celsius. That combination has made Lost City central to one of the leading hypotheses for the origin of life, developed by Michael Russell, William Martin, and Nick Lane, among others. Their argument is that the first metabolism did not require sunlight or lightning or a warm pond, but arose in the porous mineral structure of an alkaline vent, where the natural pH difference between alkaline fluid and mildly acidic ancient ocean water provided a proton gradient across thin mineral walls. That is significant because a proton gradient across a membrane is exactly how every living cell on Earth stores energy today, including yours. Chemiosmosis, worked out by Peter Mitchell in the 1960s against considerable resistance, is universal. If life began by borrowing a gradient that a rock was producing anyway, then the machinery you are running right now is a copy of a geological process. The vent field has been active for at least 30,000 years, and possibly much longer, which makes it a far more stable environment than a volcanic vent, which typically lasts decades. None of this establishes that life began there. It establishes that the necessary conditions occur naturally, persist for long periods, and are being produced right now on the floor of the Atlantic. There is a more recent and more contested claim in the same territory. In July 2024, a team led by Andrew Sweetman published a paper in Nature Geoscience reporting the detection of oxygen production in complete darkness at 4,000 meters depth on the abyssal plain, apparently associated with polymetallic nodules, and suggesting the nodules act as natural electrodes splitting seawater. They called it dark oxygen. The finding has been challenged by multiple groups, including researchers funded by deep-sea mining interests, but also independent scientists, who argue the signal could be an artifact of the measurement method. Rebuttals and responses have continued through 2025. I include it because it is a live and genuinely interesting question, and because it is a good illustration of how these discoveries actually proceed, which is contentiously and slowly. The rule is that reproduction requires sex, or at least that abandoning sex is an evolutionary dead end. The theory behind this is solid. Sexual reproduction shuffles genes between individuals, which lets beneficial mutations combine and harmful ones be purged. Without it, a lineage accumulates deleterious mutations irreversibly, a process named Muller's ratchet, and it cannot adapt fast enough to keep up with parasites, an argument called the Red Queen hypothesis. Asexual lineages in the fossil record are typically short-lived twigs. Bdelloid rotifers did not read the theory. They are microscopic animals, a few hundred microns long, found in fresh water and in mosses and lichens everywhere. There are around 460 described species. In all of them, across all the specimens ever examined, no male has ever been found. There are no males. There is no meiosis of the normal kind. Every individual is female and reproduces from unfertilized eggs. Molecular evidence indicates they have been doing this for something in the range of 40 million years, possibly longer, and during that time they have diversified into hundreds of species and colonized freshwater habitats worldwide. Olivia Judson called them an evolutionary scandal, borrowing a phrase from John Maynard Smith. Two mechanisms appear to be involved in how they get away with it. The first is desiccation. Bdelloids can dry out completely and survive, like tardigrades, and they do this routinely. Drying shatters their DNA. When they rehydrate, they repair it, and the repair process appears to allow genetic exchange between the multiple copies of their own genome, providing some of the benefit of recombination without sex. The second is theft. Genome sequencing has found that around 8% of bdelloid genes are of foreign origin, acquired horizontally from bacteria, fungi, and plants. During the repair process following desiccation, with the cell membrane compromised and DNA broken, foreign DNA present in the environment can be incorporated. Some of those acquired genes are functional and appear to be doing useful work. They also seem to use desiccation to escape parasites, since drying out kills the fungal parasites that specialize on them, while the rotifer survives, allowing an infected population to be blown away as dust and re-establish clean. In 2021, Russian researchers reported reviving a bdelloid rotifer from Siberian permafrost radiocarbon, dated to around 24,000 years old, after which it reproduced. Elsewhere in the animal kingdom, parthenogenesis appears repeatedly. Several species of whiptail lizards in the American Southwest are entirely female and reproduce clonally, and they originated from hybridization events between sexual species. They still perform courtship behavior with each other, which appears to stimulate ovulation. Then there is a group that took reproductive strategy in the opposite direction. Osedax are marine worms discovered in 2002 by researchers at the Monterey Bay Aquarium Research Institute, living on whale bones on the seafloor. The common name is bone-eating worm, and they have no mouth or gut. They bore into bone using acid-secreting root structures and rely on symbiotic bacteria to break down the lipids and collagen inside. The females are the animals you see. The males are microscopic, remain in a larval-like state their entire lives, and live inside the tube of a female, sometimes dozens or over 100 at a time, functioning as little more than sacs of sperm. A male Osedax never develops the boring structures, never feeds, and lives off the yolk it was born with. There is one more category, and it sits on the boundary of the subject rather than inside it. The rule is that viruses are simple, that they are small, and that they are not alive. That framing came from the fact that the viruses we knew about were tiny, typically 20 to 200 nanometers, small enough to pass through filters that catch bacteria, which is how they were originally defined. They carry a handful of genes, they cannot metabolize, and they cannot reproduce without a host. In 1992, during an investigation of a pneumonia outbreak in Bradford, England, researchers isolated something from a water-cooling tower that stained as a gram-positive bacterium and behaved like one. It sat in a freezer, misidentified, for over a decade. In 2003, a team in Marseille led by Didier Raoul and Bernard Lascola re-examined it and found it was a virus. They named it mimivirus, short for microbe-mimicking virus. It is about 400 nanometers across, larger than many bacteria, and visible under a light microscope. Its genome is around 1.2 million base pairs and encodes over 900 proteins, which is more genes than several parasitic bacteria have. Crucially, it carries genes nobody had ever seen in a virus, including components of the machinery for protein synthesis, specifically amino-acyl tRNA synthetases. Those genes were considered a defining feature of cellular life. Mimivirus cannot make its own proteins, so it does not translate. But it has parts of the apparatus. Then it got stranger. In 2008, the same group found a small virus infecting mimivirus itself, replicating inside the viral factory that mimivirus builds in host cells and reducing its reproductive success. They named it Sputnik and coined the term virophage. A virus with a parasite is a difficult thing to fit into the category of non-living chemical particle. In 2013, Pandora viruses were described with genomes of 2.5 million base pairs, and more than 90% of their genes had no recognizable counterpart in any known organism. In 2014, a team led by Chantal Abergel and Jean-Michel Claverie isolated a virus from Siberian permafrost, dated to around 30,000 years old, and revived it. Pithovirus sibericum is about 1.5 microns long, larger than many bacteria, and it successfully infected amoeba after 30 millennia frozen. None of these organisms metabolizes, and all of them require a host, so under most definitions they are not alive. But they have blurred the boundary enough that the debate over whether viruses belong on the tree of life has genuinely reopened, with serious proposals for a fourth domain that most virologists reject and a minority defend. In the other direction, there is Nanoarchaeum equitans, described in 2002 from a hydrothermal vent off Iceland. It is an archaeon 400 nanometers across with a genome of just under 500,000 base pairs, one of the smallest of any cellular organism. It cannot synthesize its own lipids, amino acids, or nucleotides. It lives attached to the surface of another archaeon and takes what it needs. The gap between the largest virus and the smallest cell is now smaller than the gap between the smallest and largest bacteria. The rule is that life requires a chemistry close to neutral, with moderate salt and no dissolved metals to speak of, because proteins and membranes come apart outside a narrow range. The organisms that break it are collectively called extremophiles, which is a revealing word. It means loving extremes, and the extremes in question are extreme relative to us. From the organism's point of view, these are simply the conditions it requires, and most of them die if you move them somewhere comfortable. For acid, consider the Rio Tinto in southwestern Spain, a river that runs deep red for about 100 kilometers. The color comes from dissolved iron. The pH is around 2, which is roughly the acidity of stomach acid or lemon juice, and in places it drops below 1.7. The river was long assumed to be a product of the mining that has taken place in the area since the Bronze Age. The current understanding, developed largely by Ricardo Amiles and colleagues, is that the acidity is primarily biological. Chemolithotrophic bacteria, particularly Acidithiobacillus ferrooxidans and Leptospirillum, oxidize the iron sulfide minerals in the surrounding rock and release sulfuric acid as a byproduct. The organisms are producing the conditions they live in. The river is not sterile. It supports a surprisingly diverse community, including a substantial population of eukaryotes, mostly algae and fungi, which is unexpected because eukaryotes were assumed to be less acid-tolerant than bacteria. Because of the iron chemistry and the mineral assemblage, the Rio Tinto has been used extensively as a terrestrial analog for Mars, and drilling projects there have been used specifically to test life detection instruments. At the far end of acid tolerance is Picrophyllus torridus, an archaeon isolated from a sulfataric field in northern Japan. It grows optimally at pH 0.7 and can still grow at pH 0, which is a hydrogen ion concentration around 10 million times higher than pure water. Its membrane is chemically unusual and becomes unstable if the acidity drops, meaning the organism is destroyed by neutral conditions. At the opposite end, Mono Lake in California has a pH around 10 and is nearly 3 times as salty as seawater, and it supports a dense ecosystem including brine shrimp, alkali flies, and enormous populations of migratory birds that feed on them. Salt is its own category. The Dead Sea and hypersaline evaporation ponds run at close to saturation, many times the salinity of seawater, and they are frequently colored pink by dense populations of haloarchaea. These organisms handle the osmotic problem by accumulating potassium chloride internally to match the outside concentration, which requires their entire set of proteins to be re-engineered. Their proteins carry an unusually high proportion of acidic amino acids on the surface, which keeps them soluble in brine and makes them fall apart in fresh water. Deep-sea brine pools are stranger still. On the floor of the Gulf of Mexico and the Red Sea, there are pools of hypersaline water so dense that they do not mix with the seawater above them. They have shorelines and visible surfaces, and submersibles can watch waves break on them. Around the edges, dense communities of mussels with chemosynthetic symbionts thrive. Animals that fall in are killed by the salinity and preserved, and the pools are sometimes called jacuzzis of despair for that reason. There is also a cautionary tale in this territory that belongs in any honest treatment of the subject. In 2010, a press conference was held to announce a finding that had been trailed as significant for astrobiology. A team led by Felisa Wolf-Simon reported in Science that a bacterium isolated from Mono Lake, designated GFAJ1, could substitute arsenic for phosphorus in its biological molecules, including its DNA. Phosphorus is one of the 6 elements considered essential to all known life, forming the backbone of nucleic acids and the energy currency of the cell. An organism using arsenic instead would have represented a genuinely different biochemistry. The claim collapsed within 2 years. Independent groups, notably Rosie Redfield at the University of British Columbia, and a team including Tobias Erb and Julia Vorholt, repeated the work and found that GFAJ1 is simply arsenic-tolerant and still requires phosphorus, and that the original result was best explained by trace phosphate contamination and by insufficiently rigorous purification of the DNA. Science published the rebuttals in 2012, and the original paper was later retracted. The episode is often cited as a failure, and in one sense it was. It is also a reasonably good demonstration of the process working. The claim was published, the data were made available, other laboratories tested it, the test failed, and the record was corrected in public, with much of the debate conducted openly while it was happening. The reason to include it here is that everything else in this video could, in principle, have gone the same way, and did not. Theomargarita magnifica has been examined by multiple groups. Heneguya's missing mitochondrial genome was confirmed by several independent methods. Riftia's symbionts were established beyond argument within a few years. The difference between an accepted impossible organism and a retracted one is not how strange the claim is. It is whether it survives people trying to break it. The rule is that animals are complex, with organs, tissues, symmetry, and a nervous system. The organism that breaks it can be scraped off the glass of a saltwater aquarium. Trichoplax adherens was found in 1883 in an aquarium in Austria and was assumed for decades to be the larva of something else because it seemed too simple to be an adult animal. It is a flat, irregular blob about a millimeter across, with no fixed shape, no front or back, no left or right, no mouth, no gut, no muscles, and no neurons. It has around 6 cell types. For comparison, a human has several hundred. It is the simplest known free-living animal, and it constitutes its own phylum, Placozoa. It moves by beating cilia on its lower surface and by changing shape. It feeds by settling onto a patch of algae, sealing the edges of its body against the substrate, and secreting digestive enzymes into the space underneath, effectively creating a temporary external stomach. Then it absorbs the products. It reproduces mostly by splitting in half or by budding off small fragments. For a long time, Trichoplax was assumed to be a degenerate form, an animal that had lost complexity. Its genome, published in 2008 by a team led by Mansi Srivastava and Daniel Roksar, complicated that. The genome is not simple in the way the body is. It contains a substantial number of genes associated with developmental signaling, transcription factors, and cell-to-cell communication, including many that in more complex animals are involved in building nervous systems and body axes. The toolkit is largely present. The organism just does not use it to build anything elaborate. Later work found that Trichoplax has cells that secrete peptides which coordinate behavior across the body, functioning as a chemical signaling system that produces coordinated feeding and movement without any neurons or synapses. Carolyn Smith and colleagues showed that specific peptides trigger specific behaviors, including a churning motion during feeding. It is coordination without a nervous system, in an animal, achieved by broadcasting chemicals across a body small enough that diffusion is fast enough to work. Where Placozoa sits on the animal tree is genuinely unsettled, and the argument matters. Depending on which genes and which methods are used, analyses place either sponges or comb jellies at the base of the animal tree, with placozoans nearby. The stakes are high because if comb jellies branched first, and they have neurons while sponges and placozoans do not, then either neurons evolved twice independently or they were lost twice. Neither option is comfortable, and the field has been arguing about it for over a decade. Simplicity in animals can also be secondary, meaning it evolved from complexity by loss, and the mixozoans we met earlier are the clearest case. They are cnidarians reduced from free-swimming jellyfish relatives to a few cells, retaining the stinging apparatus as an attachment device. Some mixozoan genomes are among the smallest of any animal. Dicyamids go further. They are parasites of cephalopod kidneys, consisting of roughly 20 to 40 cells in a fixed arrangement, with a single long central cell surrounded by a jacket. Whether they are simple by ancestry or by reduction was debated for a century, and molecular evidence now indicates reduction from a more complex ancestor, which means simplicity in animals is not a stage on the way to complexity. It is a destination that can be reached from either direction, and evolution has arrived there repeatedly, usually because the environment stopped rewarding the machinery. So what do all of these have in common? The obvious answer is that they are exceptions to rules. The more useful answer concerns where each one was found and when. Tardigrade extremes, characterized largely in the last 30 years. Dinococcus, 1956, in a food sterilization experiment. Strain 121, 2003, at a deep-sea vent. Halicephalus mephisto, 2011, 3 kilometers underground. Thiomargarita magnifica, 2022, in Caribbean mangroves that people walk through. Monotacomanoids, 2016. Heneguya, 2020. The third lichen partner, 2016, in an organism studied continuously since the 1860s. Riftia and the entire concept of chemosynthetic ecosystems, 1977. Desulforudis audaxviata, 2008, in a gold mine. Mimivirus, correctly identified in 2003 after 11 years in a freezer labeled as something else. Osedax, 2002. The pattern is not that biology keeps producing surprises. The pattern is that we keep looking in new places and finding that something already lives there. Every rule in this video was a generalization from a sample, and the sample was the accessible surface of one planet, examined by organisms that need moderate temperatures, oxygen, and light. We described the conditions we could survive in, catalogued what lived there, and called the result the rules of life. The deep subsurface was not searched because we assumed it was sterile. The vents were not searched because we assumed nothing could live without sunlight. Lichens were not fully sequenced because we already knew what they were made of. Thiomargarita was picked up and set down repeatedly because it was assumed to be a fungus. There is a way to quantify how skewed our attention has been. In 2018, Yinon Baron, Rob Phillips, and Ron Malo published a census of the biomass of Earth in the Proceedings of the National Academy of Sciences, and the results are worth knowing. Plants account for roughly 80% of all biomass on Earth, around 450 gigatons of carbon. Bacteria come second at around 70 gigatons, fungi at around 12, archaea at 7, protists at 4. All animals combined come to about 2 gigatons of carbon, which is under 0.5% of the total. Of that, arthropods are the largest share at 1 gigaton, fish at 7/10. All wild mammals together come to about 0.007 gigatons. Humans are about 0.06, which means we outweigh all wild mammals by roughly a factor of 10, and livestock outweighs us by a factor of 2. The organisms that get the documentaries, the ones with faces, constitute a rounding error in the living mass of this planet. And a large fraction of the microbial biomass, which is most of the biomass that is not plants, lives in the subsurface, in the sediment, in the rock, in the places we did not check until recently, and still cannot sample easily. There is a second way to measure our ignorance, which is to count the species we have not described. The most widely cited estimate for the number of eukaryotic species on Earth came from a 2011 study led by Camilo Mora, which used the way taxonomic ranks fill in over time to extrapolate a total of around 8.7 million species, with roughly 2.2 million of those in the ocean. At the time of that estimate, about 1.2 million had been formally described and catalogued, which means that even by that estimate, something in the range of 86% of species on land and 91% in the ocean had never been named. Those numbers carry large uncertainty, and other estimates run higher or lower depending on assumptions about insects, fungi, and nematodes. Fungal diversity estimates alone range from 2 to 6 million species, of which perhaps 150,000 have been described. For microbes, the situation is different in kind rather than in degree, because for most of the history of microbiology we could only study what we could grow, and the great majority of microorganisms do not grow in culture. The figure often quoted is that under 1% of environmental microbes can be cultivated with standard methods, a discrepancy known since the 1980s as the Great Plate Count anomaly. Sequencing changed, That because it lets you read the genomes of organisms directly from an environmental sample without culturing anything. When Jill Banfield's group and others began doing this systematically, they found entire branches of the tree of life that had never been observed. In 2015 and 2016, Banfield's group published revised trees of life, incorporating a huge amount of genomic data from uncultivated organisms. A large fraction of bacterial diversity turned out to sit in a group called the candidate phyla-radiation, comprising dozens of major lineages, most of which have never been seen under a microscope and are known only from assembled genomes. These organisms tend to be very small, with reduced genomes, and many appear to be dependent on other organisms in ways not yet understood. The revised tree looks nothing like the one most people were taught. Animals, plants, and fungi collapse into a small corner of one of 3 domains, and the bulk of the diversity is bacterial and archaeal lineages with no common names, no photographs, and in many cases, no cultured representative. We have been mapping a planet by describing the parts of it that we could grow on a plate or catch in a net. There is a final implication, and it is the reason this collection of oddities matters beyond the level of trivia. For most of the history of the search for life beyond Earth, the working framework has been the habitable zone. The habitable zone is defined as the range of orbital distances at which a planet could have liquid water on its surface, given the brightness of its star. Too close and the water boils. Too far and it freezes. That definition is doing a lot of unexamined work, and the word surface is the load-bearing part. Every organism in this video argues that the definition is too narrow. Life does not require sunlight, because chemosynthesis exists, and there is an entire biosphere running on rock chemistry and radioactive decay. Life does not require moderate temperature, because organisms grow at 121 degrees Celsius, and others metabolize below freezing in brine channels in sea ice. Life does not require constant water, because tardigrades and rotifers and Deinococcus can shut down entirely and wait, for years, in a state indistinguishable from death. Life does not require oxygen, and at least one animal has abandoned it completely. Life does not require a surface at all, because most of Earth's microbial life may be underground. Apply those corrections to the solar system, and the picture changes. Europa has a liquid water ocean beneath an ice shell, likely in contact with a rocky sea floor, which means the chemistry that powers vent communities is at least possible. Enceladus is actively venting water into space through fractures at its south pole, and the Cassini spacecraft flew through those plumes and detected molecular hydrogen, silica particles consistent with hydrothermal activity, and complex organic molecules. Titan has stable liquid on its surface, though it is methane and ethane rather than water, at 94 Kelvin. Mars has subsurface ice and had surface water in the past, and its subsurface may still host brines. None of that is evidence of life. It is evidence that the search area is much larger than the habitable zone definition implies, and that the reason we drew the boundary where we did was that we were describing our own requirements and mistaking them for requirements in general. Which brings this back to where we started. The title of this video is a joke at our expense. None of these organisms should not exist. Every one of them is doing exactly what the laws of physics and chemistry permit, using mechanisms that make sense once you understand the conditions they evolved in. There is nothing impossible about a bacterium that reassembles its own shattered genome or an animal that runs its development backward. The impossibility was ours. It was a statement about the limits of what we had examined, mistaken for a statement about the limits of what is. Every one of these organisms was living quietly, doing the thing we said could not be done, for millions of years before anyone arrived to declare it impossible. The tube worms had been at the vents since long before there were people to be surprised by them. Theomargarita was growing on mangrove leaves in shallow, warm, easily accessible water, visible to the naked eye for the entire history of biology as a discipline. The rules were never the problem. The rules were fine. They were accurate descriptions of the places we had been. And the useful thing to take from all of this is not a list of strange animals. It is the recognition that the phrase that should not exist has, in every single case so far, turned out to mean something considerably more modest. It has meant that nobody had looked there yet. There is a great deal of this planet where nobody has looked yet. The deep ocean below 1,000 meters constitutes the largest habitat by volume on Earth, and a small fraction of the seafloor has been mapped at high resolution, with the visually observed fraction far smaller still. The subsurface biosphere is roughly twice the volume of the oceans and is sampled through boreholes a few centimeters wide. The interior of glaciers, the sediment beneath Antarctic ice sheets, the aquifers in continental basement rock, the pore spaces in ordinary soil at depth, none of these have been surveyed in any systematic way. Every organism in this video came out of a place like that, and in most cases the discovery was made by 1 or 2 people who went and looked because nobody had. The tardigrade was found by a country pastor with a microscope. Thiomargarita was picked up by a researcher who kept noticing white filaments on mangrove leaves. The third lichen partner was found by a graduate student who refused to accept that 2 identical-looking organisms could behave differently for no reason. Riftia was found because a geology expedition happened to point a camera at the wrong thing. None of it required new physics. It required going somewhere and paying attention. Which is the closing thought worth carrying, because the list in this video is not finished, and there is no reason to think it ever will be. Somewhere on this planet, right now, in a place that is currently classified as uninhabitable and therefore unexamined, something is alive and doing a thing we have written down as impossible. It has been there the entire time. It is not hiding. We simply have not been.